Good morning. My name is Dr. Karen Carlton. I'm a veterinarian and a scientist here at the University of Guelph, completing my doctoral program in pathology, which means the study of disease. As part of Science Literacy Week, I've been asked to make a little video highlighting kind of a day in the life of a scientist here at the University of Guelph. So I'm hoping to take you into the Department of Pathobiology so that you can see what my lab looks like and see what kind of things I do in my day. Now, I'm a vaccine researcher. I don't work on COVID, but I do work on another virus that infects sheep, as well as infecting goats and people and cows and ticks. And so my research is looking at a vaccine that we can give to these sheep. And we're trying to answer two main questions that are very important as part of early vaccine development. And those are, do we think the vaccine is safe? So can we prove that there is very little risk of giving the vaccine with huge potential benefit? And two, does the vaccine work? So can we prove that the sheep's immune system, after they're vaccinated, is better equipped or trained to fight off the virus in the future? So let's head inside. I've just entered the lab in the morning. I've come in early to give the tour, so I wouldn't disrupt anyone, so I'm the only one in here right now, but you can see this is a very large lab. So this is a shared space. There are a few different primary investigators that share this lab and they share the equipment. So as we tour around, you can see there's all sorts of fancy pieces of equipment in here. Some of it is for working on DNA analysis. Some of it is for working on viral projects. Some of it works on cancer. We've got a whole set of fridges and freezers, so we're able to store our samples at an appropriate temperature. All of the researchers and scientists in here have their own little workspace with all of the specialized equipment they need to do their projects. So right here we have one of the most important pieces of equipment I use for my research, called a centrifuge. A centrifuge has a giant rotor inside that can spin samples very quickly, like hundreds of times per second. So I'm able to put my blood samples in here. It's going to spin around extremely quickly, and that's going to allow for the different parts of the blood to separate out from each other. So I'm going to show you a picture here. This is just an example I found on the internet. The tube goes in and it's all filled with blood, and when it comes out of the machine, it is separated by components. So at the top, this yellow here, we have our serum. That contains our antibodies, which are very important. That's what we want to generate using the vaccine. At the bottom, we have all of our red blood cells, and in the middle, we have something called the Buffy coat. And the Buffy coat contains our white blood cells. So in addition to sampling the serum for antibodies to see if the vaccine worked, we're going to do something cool with the Buffy coat. So I'll show you that next. So once we've collected our Buffy coat in a specialized way, I'm able to apply antibodies to the white blood cells and then run them through this machine here called the flow cytometer. Basically, the antibody-labeled cells run one by one by one by one through the machine in single file line, where they are exposed to a laser beam, and then depending on what color light the cells emit, they can be sorted into different cell populations. By comparing those cell populations before the vaccine versus after the vaccine is one of the ways that we can assess whether or not we think the vaccine is working. Operating this machine is going to take probably the rest of my morning, so we'll check back in this afternoon. All right, so we're back after lunch. After assessing our antibody levels and looking at the various levels of white blood cells, the next step is to determine whether or not this vaccine is safe. So one of the ways that we do this is we look at biopsies of the vaccine site under a microscope. So I'm here in the scope room. So we're in a different room now, apart from the lab. And what we're going to do is we're going to look at biopsies from some of our vaccine sites. As part of this study, we administered the vaccine to two different groups of sheep in two different ways. The first group got an injection into their muscle. So similar to how we get a flu shot in our arm, we administered the vaccine into a muscle belly, but on the sheep, we chose the back of the left leg, just beside the tail. The second group of sheep got a tattoo vaccine, which I think is really cool because we don't really see this done in humans. Administering a vaccine using a tattoo has two main advantages. The first is it allows for visual identification that an animal has been vaccinated. So in our case, we administered the tattoo to the inside of the sheep's ear. thinking this would allow a farmer to see the sheep from a distance and be able to tell if it was vaccinated. The second big advantage of tattoo vaccines is that it prevents us from having to put a needle into a potentially expensive cut of meat, as sheep are agricultural animals that are consumed for food. So when I take those slides and I look at them under the microscope, the types of things I'm looking for is evidence of damage. I'm looking for areas where there's bleeding, I'm looking for inflammatory cells, and I'm looking for swelling or edema. So this is a live image of what I'm looking at under the microscope right now. And this is a biopsy taken from one of the sheep's ears about 48 hours after it was vaccinated. The outside of the ear is hairy. So these little dots are little hair follicles. The middle of the ear has cartilage, just like the ears of people. And the inside of the ear, we don't see any hair follicles. So this is where we administered the tattoo. So basically what I do is I scan around on the microscope and I'm looking, hopefully you can see my little pointer there. I'm looking for areas with bleeding or inflammation or swelling on the inside of the ear. And I'm also looking for evidence of the actual vaccine. So it'll be kind of a hazy light blue color to tell me that it made it into where it should go. So I'm going to spend about the next 2 hours or so looking at hopefully about 25 different biopsy sites and recording how much damage there is. Hopefully we don't find any because we want our vaccine to be safe as possible. And the other thing that I'm going to spend some time doing is looking at the blood work of the sheep. So in addition to taking blood so that we can look at antibodies and white blood cells, we're able to perform testing to make sure all of the red blood cells are happy and that we don't have any enzyme changes that could indicate to us that there's been some sort of internal damage with, let's say, the kidney or the liver. So lots of work to do this afternoon. Check in a bit. All right, so after looking at about 20 slides or so, my eyes are starting to get a little bit tired, so it's time I move on to my last task of the day, and it's about 3 p.m., and that's working on my lectures for this fall. As a scientist, I think it's really important that we share knowledge with others, and so I elect to teach in both virology and toxicology for the second-year veterinary students. So thank you so much for joining me today as a kind of little, what does a scientist do? What does my day look like as a scientist here at the University of Guelph? I appreciate you taking the time to watch this video, and I hope you enjoy the rest of the Science Literacy Week programming. Thanks again for joining me. Stay safe in these weird times. Take care.